Okay, this is 5B. This is going to talk about photosynthesis and cellular respiration. 5A was the actual equations. This one's going to be the actual organelles. In years past, I've asked my kids to draw this, and this year I gave it to them on their PowerPoint slides. But I've got this large chloroplast that has an outside membrane called the outer membrane and an inside membrane called the inner membrane. This is a point of evidence that we've used to actually bring in a prokaryotic cell into a eukaryotic cell. And what happened was that this thing did a really good job of taking sunshine along with carbon dioxide and water and producing glucose. So the prokaryotic cell was invited into the eukaryotic cell, saying, I'll give you everything you need. All you got to do for me is give me the glucose. So if you take a look at it inside this chloroplast, inside these two membranes, we've got this jelly-filled area, sort of like cytosol, with these coins stacked up. I'm going to call them thin mints, but these are called the grana. So this is a stack. The stack is called grana. There's another stack, grana. There's another grana. There's a grana. Here's a grana. Everywhere a grana, grana. But these grana actually are made of these individual sections. The inside, the minty section, that's the cream mint, or the lumen. That's the thylakoid space, the actual space in between the individual coins, which are called the thylakoids. And this is the thylakoid membrane, sort of the chocolate coating around the thin mints, the mint cream on the inside, which makes up a big stack of them for the thin mints. And then the last thing is we have the cytoplasm, that's the stroma. And the cytoplasm, of this prokaryotic chloroplast machine, which is going to be take some of the control of the photosynthesis. Okay, the next slide actually showed a real picture of a highly magnified chloroplast. You can see the grana, you can maybe see some of the thylakoids, you can definitely see the stroma. Two membranes, a little tough to see, but they're out there somewhere. Here's another large view talking about the grana and the thylakoids. This one actually shows that it has ribosomes. Remember, ribosomes make proteins, and with the DNA, the circular prokaryotic DNA, chloroplasts are in charge of their own mitosis. So we have the lumen on the inside, the thylakoid membrane, we have the grana, stromas out there, there's the DNA, the granum, individual one, we have the external membrane and the internal membrane and the internal membrane space. We even have plastic globules, which I'm not exactly sure what they do. Okay, the next thing is I had them draw mitochondria in years past, but I gave it to them this year as well. Mitochondria also come with an outer membrane and an inner membrane, and they have a space in between, which is called the intermembrane. And then we have these large, like curves, almost like somebody takes a blanket or a sheet and curls it up. These increase surface area. These are the cristae, which are the folds, and then the inside, which has got a jelly-type material in it, which you'll see on the next slide. That stuff's called the matrix, and I'm going to call that the jelly inside. And these are going to be the things that are going to be in charge of taking the glucose and turning it into ATP. Okay, what you see here is a real mitochondrion. You can see the cristae that are inside. You can actually make up the matrix. Can't really see the two membranes. See other organelles that are out on the inside. Looks like a rough endoplasmic reticula, but this is going to be in charge of the ATP. And here's a color version. Shows the jelly inside for the matrix, the cristae, which is the increasing the surface area, the folds that are inside, the outer membrane, the inner membrane, and then the space in between. So then we're going to go back to photosynthesis, and I'm going to ask them to draw again, but of course I gave this to them. This is a chloroplast, where we actually have the sun on one side and we have night on the other side. When I grew up, these were called the light reactions. These guys were called the dark reactions, but now we're going to be calling them light dependent versus light independent. Side that needs light, side that does not need the light. And that sort of was a misnomer way in the back when I was learning it, is that we thought this would happen in the daytime and we thought this would happen in the nighttime. This really does happen in the daytime, but this happens 24/7. So if you take a look at the picture, we get sunlight coming in, they're going to hit the grana, and this is called the light reaction, and with the process with the water coming in. There it is. Light and water. This side actually gives the oxygen, which you guys know in photosynthesis is part of the products of photosynthesis that they create for themselves and the animals. On the dark side, it also produces ATP and an electron carrier called NADPH. And then on the dark side, which happens all the time, when this ATP goes over to the Kelvin cycle and the NADPH goes over to the Kelvin cycle, this is a highly cyclic enzymatic reaction. This is where we pull in the carbon dioxide if we're a plant, and we use these, the power and the electrons to make the sugar, the C6H12O6 glucose. So this is the light-dependent part of it. This is the light-independent part of it. And you can see there it goes. And then there's the light-dependent side that requires light and water to produce oxygen. Here's the light-independent, the side that actually absorbs carbon dioxide and produces the sugar. Light-dependent reactions, again, same sort of picture. Light goes in, water goes in. So this happens in the thylakoid membrane, actually inside those little coins, those thin mints of the grana. It uses light energy to make ATP, and you can see the ATP going off here, plus it excites electrons in the electron carrier for the NADPH. It also produces the oxygen, takes the water and actually splits it, hydrolysis, and actually splits off the O2 and actually puts that up in the atmosphere. Actually escapes the chloroplast from the chlorophyll. NADPH, the electron carrier, it gets released into the actual cytoplasm, the stroma, and ATP goes into the stroma. And then we go to the light-dependent side, and remember this one happens 24/7. This one requires light. And enzyme reactions in the stroma, carbon dioxide comes in. The energy from the light-dependent reactions are used to make the glucose. So we've got this light-dependent material coming in, and we're going to make glucose. The ATP and the changes to ADP, and the NADPH changes into NADPH plus, and they're going to go back and get re-energized to go back into ATP, and this is that ATP, ADP, and actually also the NADPH, NADP plus cycle that goes on inside of a chloroplast. This is also called the Kelvin cycle because the scientist with the last name Kelvin actually identified all the enzymatic reactions that go in there. And in AP biology and in college you'll actually learn all these reactions, and some of those things are incredibly difficult to pronounce. Reminders that photosynthesis, we take in water, we take in carbon dioxide as our products, and we give off glucose and oxygen as the products. Here's that balanced equation again using stoichiometry, where we need 6 carbons, 12 hydrogens, 6 oxygens, and 12 oxygens. So we need to put a 6 in here to get the 6 carbons, 6 here to get the 12 hydrogens, 6 there to get 6 oxygens, and then 6 there to get 12 oxygens. And then we're going to take the light-dependent and light-independent reaction, and we're going to talk about where it happens in terms of photosynthesis and also the Kelvin cycle, and the photosynthesis again requiring light, and Kelvin cycle 24/7. So this happens in the thylakoid membranes, and what we need is we need sunshine as our energy source. We need ADP to move in, which we're going to energize, and we need the NADP plus, which we're also going to energize, and we need to bring in water. And what we get for a product, we get ATP energizing the ADP with another phosphate, putting the energy back in. We change the NADPH and DP plus into NADPH, and then instead of the water we actually lyse it and make oxygen. And then we have the light dependent, and this one happens out in the stroma, the cytoplasm of the chlorophyll. And this side we need the ATP, we need the NADPH, and we need to bring in carbon dioxide from the atmosphere, and what we make over here is glucose, C6H12O6. So this is a different way of looking at the last diagram, maybe a little more clean, but I asked my kids to fill it in. And there's also a shock wave down here on photosynthesis, which you're welcome to watch if you wish. Then reactions of photosynthesis, we actually do have some limiting factors. First one being water. You cut off water from a a cell and we turn it into a hypotonic reaction, where the hypotonic goes out into the hypertonic solution and the water actually goes in and the plant wilts and eventually may actually kill the plant, but certainly would slow down photosynthesis. Temperature, you guys all know that enzymatic reactions need certain temperatures. Plants, just like animals, have to have a temperature probably between 0 and 35 degrees Celsius, which is pretty darn warm. Your body's 37. And temperatures above that or below that are going to damage the enzymes, and since there are enzymatic reactions going on, photosynthesis slows down or stops. And the last thing is light intensity. We actually give a plant light and it does what it's supposed to do. I guess theoretically if you give it too much light it could burn. Don't give it enough light, and I know some of you have done this experiment in second grade for a science fair project to find the light reaction on plants. You put one plant out in the sun, you put another plant in a closet, and you find out the one in the closet doesn't do as well. And the last one is color, and I like to put that little smiley face there because I got this from a British or maybe a Canadian site. And color, Roy G. Biv, which the colors which we can see from our eye, really it's red, yellow, and blue, and the other colors are just mixtures, but it's really the red and the blue-violet that the plants like. And if you think about a plant being green, that's the color that it doesn't like, so it doesn't really care for the green and the yellow. It likes the two other ends. Okay, a food lesson. There's a lot of energy stored in food. You take a gram of sugar, glucose, C6H12O6, and that has 3,800 plus calories of heat. In science, we learned that a calorie is the amount of energy that we need to raise the temperature of 1 gram of water 1 degree Celsius. So this could actually take the amount of water, 3.811 liters, and raise it 1 degree Celsius, or it could take 1 gram of water and raise it 3,811 degrees Celsius. In other words, definitely turn it into vapor very easily. But in food, we don't use these calories. We use something that's called kilocalories, 1,000 calories. And what that means is 1 gram of sugar instead of actually holding 3,811 is now 3.811 food calories. And they do this to make food sound like it doesn't have as much energy. I'm not exactly sure if people would take like this candy bar that may have 250 calories when it really has 250,000 calories. So when you look at the calories right there, this particular product that has 7 grams of fat, 4 grams of bad fat, has a pretty good amount of sodium, has a lot of carbohydrates, sugars, 20, not real good carbohydrate production. It does have 2 grams of protein, but it has 180,000 temperature calories, science calories, or 180 food calories, because remember these are kilocalories. Last section we're going to talk about is cellular respiration, and here's a picture I found on Facebook a few days ago, and what it says: in a 24-hour period your body will blink 23,000 plus times, your heart will pump 100,000 times, you'll breathe 23,000 times, your liver will filter 1,500 liters of blood. That's little, you divide it by 4 and you can figure out gallons, but that's a lot of liquid. And your stomach produces 2 liters of acid. So how does your body actually do this? What does it use as the energy? Glucose is sugar, but your body can't utilize the sugar. But what you do need is you need that cellular respiration effect on the cristae and the stroma, the matrix of the mitochondria. So 3 steps of cellular respiration. Here's a mitochondria, and you can see the first one, glycolysis, happens out here in the cytosol, in the cytoplasm. This is where we take a glucose molecule, C6H12O6, and we break it down into what are called 2 pyruvates, and we also produce NADH, which is an electron carrier, which is going to move over, and you can see it drops down here in our last electron transport chain. So this occurs with or without oxygen, so you can do this in an aerobic or an anaerobic environment. This is something that all prokaryotic cells do for their ATP production. So we're going to make 2 ATPs. This is a pretty decent amount of energy production, but it's really nothing compared to the fact that if we get a mitochondrion, and remember this is maybe a prokaryotic cell that did cellular respiration really well, that eukaryotic cells invited into. Again, it's got its own DNA, it's got two membranes, it's got its own ribosomes. It's almost like it's its own little creature inside of every one of our cells. And it's also something that you can actually produce more of if you actually do some exercise. So if you're out there exercising hard, your body will start producing more mitochondria because you're going to need more ATP to do the things that you need to do. Okay, so we get two ATPs and we make an electron carrier that's going to go over here to the electron transport, and then we move over to what's called the Krebs cycle after Dr. Krebs, and this is going to happen inside the mitochondria. It's going to be in the matrix, so it's in that jelly section that's inside. And with this one, it's a series of enzymatic reactions where we're going to take the pyruvate that moved in from glycolysis, and it's going to produce 4 ATPs. It's going to produce some NADH and some FADH2, which are electron carriers. So you can see it comes in from glycolysis. We get some more from the Krebs cycles. We pick up double the amount of ATP, and this is one advantage of being a prokaryotic cell. If you have mitochondria, you can actually get 3 times as much ATP production. But then the real kicker is when we move into the electron transport chain. This is located in the inner membrane, so this will happen in between the two membranes inside the cristae, which is the part where we actually made larger surface areas so we can have more area for this to happen. Series of transport proteins. This is where we're going to take that NADH and the FADH2, and we're going to actually use them to release energy, and that energy is going to get transformed into 32 ATPs. Now this is a prime example. A lot of times the mitochondria don't make quite as many as this, but this is where it really, if it was running at 100% efficiency, this is what we would get. And then we also take the oxygen and we turn it back into water. Remember, photosynthesis takes water and turns it into oxygen. This is exactly the opposite. And then we're going to actually move these things into other materials as well, and it'll get kicked back into this system so we can start it all over again. Steps 1 through 3 takes one glucose molecule, turns it into pyruvate, turns it into electron carriers. The Krebs cycle turns it into more electron carriers and ATP, and it can make up to 38 ATPs. Prokaryotes 2, eukaryotes up to 38. So this is like 18 times more energy efficient just by having mitochondria. Probably why prokaryotic cells have not been changed as much as eukaryotic cells. We just have more chances, more energy to evolve and to change, migrate into different things. Larger animals, faster animals, warm-blooded animals, having multi-celled creatures. Reminders, cellular respiration takes glucose and water as their reactants and produces water and carbon dioxide and ATP as their products. Same sort of a data table. Here's that stoichiometry, that balanced equation, where we're going to take that plus the 6 oxygens and 6 waters to make 6 carbon dioxides, and 12 waters plus up to 38 ATPs. This happens in the cytosol, happens with air or without air, aerobic or anaerobic. What it produces, what it takes in is glucose, and what it produces is pyruvates, 2 ATPs, and NADPH. Mitochondrion, this is going to now be in the matrix of the mitochondria. The pyruvate moves into the matrix, so the pyruvate is the reactant, and what we get is 4, so we actually triple the ATPs, and then we make CO2. And then in the mitochondria in the inner membrane, remember the cristae with the surface area increase, we're going to take the NADH from up here, we're going to take the FADH2 from here, and we're going to take oxygen, and what we're going to produce is 32 more ATPs for a grand total of up to 38 if we have both of these. And all cells again can do glycolysis, including yours. This is the only way prokaryotic cells actually get their ATP, and with the help of mitochondria, which only eukaryotic cells have, we can make 18 times more ATP. And here's another shock wave you can watch if you wish. I think that actually makes the end. I'm sorry, we get water in that case too, because we got to take the water back for the chloroplast so we can make oxygen. So we take the oxygen, we turn it back into water. We take glucose and make it into pyruvate. We take the pyruvate and make it ATP and CO2 plus NADH and FADH2. Those go down here in the inner membrane for the reactants of the electron transport chain, and with the help of a whole lot of different enzymatic reactions, we make a bunch of ATP plus water. And I think that's the end of the section. I'm going to do this. Yep, that is 5C, which we'll talk about in the next video. And let's go back to here, and let's stop this, and hope to see you soon.